Good afternoon, everyone. We'll get started. So, welcome to the Sanford Consortium for Regenerative Medicine. I'm stepping in for Donna Scarrett, who couldn't be here today. I hope everyone is enjoying the conference as much as I am. It really is one of the best of the year. The presentations, the panel discussions especially, are superb. As we transition into the scientific focus of the meeting, we are very pleased to bring you a leader in the tissue engineering space, Dr. Jennifer Elisseeff, who is a professor and director of the Translational Tissue Engineering Center at Johns Hopkins, as well as the Jules Stein Chair in Ophthalmology at Wilmer Eye Institute. After obtaining her Ph.D. in medical engineering from the Harvard-MIT Division of Health Sciences and Technology, she was a fellow at the National Institute of General Medical Sciences Pharmacology Research Associate Program, where she worked in the National Institute of Dental and Craniofacial Research. She became assistant professor in 2001 and was elected as a fellow to the American Institute for Medical and Biological Engineering and a Young Global Leader in the World Economic Forum in 2008. She had co-founded Cartilix in 2004, which translated adhesive and biomaterial technologies for treating orthopedic disease, which was acquired by Biomet in 2009. She then founded Ageria Soft Tissue and Tissue Repair, startups focused on soft tissue regeneration and wound healing. She serves on the scientific advisory boards for Bausch & Lomb, Kaiser Biopharmaceutical, and Cellular Bioengineering. She has received numerous awards, including the Carnegie Mellon Young Alumni Award and the Arthritis Investigator Award from the Arthritis Foundation. She was named as a top innovator under 35 by Technology Review magazine in 2002, as well as the top 10 technologies to change the future. Tissue engineering R&D has been ongoing for decades. The clinical applications are becoming more and more of a reality, such as in the areas of tissue and soft tissue and cartilage repair, areas in which Dr. Elisseeff has been an innovator. Please join me in welcoming Dr. Jennifer Elisseeff, scientist, engineer, educator, awardee, and entrepreneur, to help us understand the promise and status of regenerative medicine from her expert perspective and projects. Jennifer, pass the podium to you.
Thank you for such a lovely introduction, and thank you for coming here tonight and spending a little time before the drinks. My goal is to time it so we'll hit sunset right when we go out. I just checked the timing for the sunset. So you and me, we're made up of, or we're defined by our genes and our environment. If you think of the whole development process, it's quite impressive. We can run from one single cell that's growing and dividing, and we've learned lots of things from that development process. We've gained things like embryonic stem cells at certain stages, and really understanding how can that one cell proliferate and differentiate and move around and make all of these tissues in the body. The ultimate endpoint being a complex infant. Right? This baby, though, is still undifferentiated, right? It's got all the DNA there, but there's still a lot of differentiation to happen. That's going to be defined by a lot of different things, right? Are they going to grow up with a beautiful beach outside, or in the city, or in the mountains? What kind of food will be eaten? What kind of family will they be blessed or cursed with? That constant question of nature versus nurture. Now, there's some great examples in nature with regeneration. The salamander limb is always sort of the poster animal for regeneration. They can regrow an arm that has all these different tissues, and you know, if we could even think of that, growing an arm where you have skin, muscle, nerve, so many different pieces. Well, if we look down to the basics of the tissue in this individual, we can have that same question of nature versus nurture. So we have the cell. It gets a lot of credit, right? It's the living piece, and it's producing all that tissue. But the surrounding environment of that cell is also very critical. And they talk to each other. Yes, of course, the cell does produce that matrix and communicates with the environment, but also that environment communicates with the cell and gives instructions, maybe even has a history of what happened in that environment. There's a dynamic reciprocity happening. We can use that same concept of nature and nurture when we think of repair. So tissue repair in the body, we have a cellular building block component, and then we have a scaffold component. And that scaffold component is what I'll be talking about most of the lecture. Now, if we look at the history of biomaterials in the body, when we think of scaffolds, it's actually relatively long. So in the mid-20th century is when a lot of these devices came into use clinically. And if you look at all these materials, you see a lot of metals and plastics, and these materials were generally designed to be hidden from the body. Actually, there were usually materials that were off the shelf for other applications that were then taken for clinical application. As we're moving towards more biological interactions and creating an environment that actually does communicate with cells and nurtures regeneration, we need to think a little bit more biologically and talk about materials that can really be seen by the body, not hidden, and give instructions that are going to help with the regeneration process. So we often think of it as a scaffold design where we have two components. We have a synthetic component that can be highly controlled chemistry, right? So depending on where we're going to implant it and how we're going to implant it, we can play around with that chemistry. Oftentimes, we don't always have the greatest biointeraction with those types of synthetic materials. So then we can incorporate a natural component. This can be peptides, proteins, proteoglycans, or a mixture of them. Essentially, very biocompatible materials that can give a lot of instruction to cells. Now, what kind of cell am I talking about here? This will change throughout the talk, but right now, think of it as a cell that's native to the tissue, a fully differentiated cell, or that you might extract from a patient, or some type of stem cell. Now, if we look at the general classes of these scaffolds, we also have a number of choices. We can have hydrogel-like materials that can encapsulate cells and suspend them. And we can have more solid-like materials. The fibers shown here, or you can think of sponges, is something essentially that a cell can really grab onto and hold. Depending on your tissue-targeted application and the clinical practice, you can choose one or the other, or even a combination of them for your application. So when we think of how we approach the whole process of regenerative medicine and translation, we think of creating those environments where we can study cell material interactions in a dish, learn about how they talk to each other. Then when we think of translation, most of the discussions of translation that I'll talk about today are how we can take those scaffolds then to modulate endogenous cells. Right? So can we capture the stem cells that are already there in your body, or progenitor cells, or some system that's already there in your normal healing process, and guide it to be more efficient and more productive? So we can learn how these environments can help teach us about what happens in the body. And this iterative process with these dotted lines is really critical. One of the most challenging things is trying to understand how do these in vitro results, structure function principles that we learn, how do they correlate that to what happens in even preclinical environments in animal models, and does that actually relate to things that happen in people? So often, we're spending time looking at things in a dish that are actually not the most important therapeutic factors. So can we understand what is really important therapeutically in what we're designing for regenerative medicine? Now, I'm not going to be talking much about stem cells, but since the conference does have stem cells in the title, I thought I should put one slide. So here's the one slide. So we did do a fair amount of work looking at how embryonic stem cells can be cultured and grown into various tissues using biomaterial scaffolds. And we were focusing, as mentioned, down sort of mesodermal type tissues with cartilage and bone and adipose. And a lot of people initially thought we were crazy. You have adult stem cells that can do those things, why even bother? So we spent a lot of time trying to understand first how to make those tissues and understanding how embryonic cells were very robust at building those tissues. And about that time, those clinical trials on the adult stem cells were happening and starting to become apparent that they weren't actually operating therapeutically how they were being studied in a dish. In a dish, they were being studied at how we can build these different tissues, but therapeutically, they were secreting factors that were really important. So again, we could study that in a dish, but it also gave us this clue that, well, what happens therapeutically is often not how we've originally designed things. So now towards clinical application, we're going to start with the knee. So this is your knee. We've got nice cartilage here on the surface of the articulating joints. But there are a lot of things that can go wrong. So I've put a number of defects in here. You can take your pick. This is a nice traumatic defect, usually a sports injury or some type of traumatic defect. And then I've shown some fibrillated tissue here. This is like arthritis and a generalized degradation of the tissue. I've even torn a meniscus here, and I've shown bony changes that happen here. Right? These are all things that lead someone to come in to their doctor and say, my knee hurts. So the first place we started was looking at scaffolds for rebuilding and filling up that hole. And I'll talk about how we took that clinically. But then after that experience of clinical translation, we realized it's not just as simple as making the tissue, filling that hole. You need to do a lot more. We need to deal with inflammation in the joint. Right? So do drugs need to be given in addition to this? We can't put a nice piece of tissue in a very unsupportive environment. Another piece that's important in the joint is the synovial fluid. Right? So you can, again, put a perfect tissue in there, but if it's not lubricated like normal articular cartilage, it's not going to survive, and the patient will still have pain. So we first started looking at the normal environment, that normal environment that the chondrocyte lives in, right? So what does it see? And we can take each of those little pieces of what it sees and put it into a scaffold. And this is just a picture of a cell in a purely synthetic scaffold. And then as we add different biology in there, you can see how the cell shape changes, and this correlates then with gene expression changes and differences in the ability to produce tissue. I'd like to talk about one example in particular, this chondroitin sulfate, which you might have heard of, various supplements. And actually, Shiny Varghese, who's here at UCSD now, worked on this and designing these synthetic biological composite materials. And what we noticed just in the dish is you saw these big blobs of cartilage forming in the dish. So this looks nice. And as we look closer, you can see these cells clustering together. And this reminded us of some things that happen in normal tissue development. So when your limb is normally developing, you get this condensation of the mesenchyme, where you get these cell-cell contacts shown here in green, these N-cadherins, and then after that you get an early transcription factor, SOX9, and then you get tissue development. So this is going back to that embryological development I showed early on, and ideally we'd like to capture some elements of that in our regeneration strategies. So lo and behold, when we had that chondroitin sulfate polyethylene glycol synthetic biological composite, we saw those N-cadherins go up, followed by SOX9 transcription, and we got better cartilage. So that's very exciting, but then what do you do? That's building it in a dish. So then we put the surgeon and the chemist together to design a solution to deal with that challenge of going from the bench to the bedside. And they took that chondroitin sulfate molecule and they made two modifications. The first one was to do this little ring opening so we could react it with tissue. And the second modification was to react with our hydrogel that we put on there. So it's like a tissue primer, or you can think of it as a paint primer. So a paint primer helps bond the paint to the surface of the wall. It's the same way the tissue primer helps bind this hydrogel that we put in there to the tissue surface. So this is a little picture of a hydrogel with a little piece of cartilage. Now again, how do we take this clinically? Well, as I showed those stem cells early on, and we definitely studied those in a dish, but when it came to translation, we had to do something a little simpler. And I thought that experience of translation and efficient translation was key to understanding how biomaterials work for regeneration applications. So we looked at a standard surgical technique that was being used frequently at the time, microfracture. And essentially, it's taking a little drill or a little hammer and just digging it down about 3 millimeters into the bone so you get some bleeding that happens. And you get this little thin piece of cartilage, more like a scar cartilage that forms. But you have cells migrating from the marrow, and you have blood and bone marrow trapped in that space. So could we use that as our cellular building block in tissue repair? This is a picture of the initial clinical procedure. So you have your defect here, clean it up, do your microfracture, paint on the adhesive, add your hydrogel, and then you've got bleeding happening in the defect. So this is an example of some of our early patients. This is the microfracture holes, and then after we implanted the hydrogel, you can see it's opaque. And this patient, actually, the tourniquet that holds, prevents the blood from going into the defect during surgery broke. And so we got to see the hydrogel fill nicely with blood, which is what I was actually happy about. So we call this also the hockey puck defect. And that's useful for two reasons. One, we have this nice cartilage around here that's really useful for crosstalk and repair in that defect. And you don't have to take away all that good cartilage. You can keep as much of the good cartilage and form your material and defect in situ. And allow it to take the shape of that defect. We used a light to polymerize this after it's implanted. Now, I'm afraid I've only have some of the early data from just the first small cohort of patients. So with the implant, we get defect fill. This is 12 months. It's now gone up to 2 years and is stable. Now I just have early pain data, and what we see is decreased pain severity and frequency with the implants. This has been further expanded by Biomet, and I don't quite have permission to give you all the data, but I can tell you it looks nice. So we're excited about that moving forward. Now, I want to get back to the lubrication because I think that's a nice story and it's very sort of commercially relevant. So I took this off the internet. So you have a patient that comes in with this red, painful joint. You inject hyaluronic acid, and here it's nice and blue and cool. Is this really true? Well, synovial fluid is sort of the oil for your joint, and it's supposed to decrease the friction. But when you inject this, why would it stay around very long, right? It's going to diffuse out pretty quickly. This is actually a problem in a lot of industry, and it's always fun to mix different fields. And I like the example of the car engine best, because I think it's something we probably experience, all of us. You might have noticed that you have to change the oil in your car less frequently than you used to, right? Part of that is synthetic oils that are more efficient. But the other reason is because the people designing those oils and the people designing the surface of those engines started working together so they could be designed to work synergistically. And that's how your body works. So normally on your cartilage tissue surface, you have proteins just on the surface there that are designed to interact with that synovial fluid or certain components in that synovial fluid. Unfortunately, when you have cartilage loss or arthritis, you lose those proteins. So what we did is we replaced them with sort of biosynthetic technology, where we have a synthetic polymer here and a peptide that we actually stole from the cancer literature. Right? It was designed to prevent CD44-mediated cancer metastasis. We took it, put on a polymer to bind hyaluronic acid to the tissue surface. So I have an artistic...
Osteoarthritis, or OA, is the most common joint disorder due to aging, wear and tear on a joint, and other factors. Here at Johns Hopkins University, we're trying to decrease the pain caused by OA in articular joints.
So you have the cartilage...
Our study starts with taking a biopsy sample from an OA knee. By taking advantage of the molecular structure of the cartilage, we attach a biomaterial known as polyethylene glycol. A second material, a peptide, is then attached. The sample is submerged in lubricant, allowing it to adhere to the prepared surface. Testing has shown that friction is reduced and further wear and tear is prevented, making this a step forward in OA treatment.
And a happy patient. I couldn't pass that one up. So, you know, once again, we had to put the surgeon and the chemist together because we could do that in a dish and show that we could reduce friction and cartilage, but then how do you put it in a one-step simple injection? What we did is we took one peptide there that could bind the tissue surface, collagens, and then the other side that could bind that hyaluronic acid. And this essentially helps keep the hyaluronic acid where it should be. So you can take cartilage, treat it with this binding peptide, and have the hyaluronic acid on the tissue surface, and have the same lubrication properties as if you were in a full bath of hyaluronic acid. right? And that makes sense because your knee joint is not going to have a full bath of hyaluronic acid. It's just going to have it targeted where it's needed to do its job of reducing friction. We're now applying this to arthritis disease models. So we've been working on cartilage for a while, and an ophthalmologist walks in the office and says, well, cartilage is nice, but you know, the eye is so much more important. People care more about their sight than they do being able to walk around. So we started applying some of these same strategies that we used for cartilage to applications in the eye. We worked with the applied physics lab of Johns Hopkins and the military to try to design an eye patch. And I'm not going to talk too much about this, but I just wanted to show you something that I'm really excited about, is that our latest example is that we can build this cornea structure purely from collagen and proteoglycans that mimic aspects of normal corneal development. And this isn't easy. So this is a native cornea, and collagen fibers come out, and then they go across, and they stack up in this way that makes it clear. Right? So it's mostly collagen, just like your skin, but it's clear because it's organized in this way. And by following the principles of developmental biology, we can create a mimic that actually looks pretty clear. And we're working with a company in Hawaii, always good to work with a company in Hawaii, translating this technology. Now back to this region. I'm not going to talk about the eye, but I'm going to talk about the craniofacial region. And I often joke that this was our dark side project. So this was a project that we did with Kythera Biopharma, which has been in the news recently. They just completed a nice acquisition. But this was the dark side project not because it was in Los Angeles, but because it was dealing with aesthetic applications. I thought, well, what do we really want to do in an academic lab working on aesthetic applications? So they were interested in taking fillers, so hyaluronic acid again, that I just talked about. And I assume everyone knows what it is. So you inject it under a wrinkle and you puff it out, and so you don't see the wrinkle as much anymore. But it goes away, just like it did in the joint. So just like we used those light polymerizations in the joint, they wanted to use light over the skin to solidify it in place. So we built a whole library of materials that allowed them to do that. I won't go into the details of that, but I'll just jump to the important part, is we got to do something really cool. So the smart company they are, they said, okay, we'll give you a free tummy tuck to a couple of people in Canada. We'll give you a free tummy tuck, but we get to inject something in a couple months before, right? And well, for the actual data, so here's the implant that's photopolymerized, that's cross-linked, so it stays longer. And this is after 3 months, and you can see how by MRI, you can see this is, has stayed in place and kept its volume, and then just the plain filler has started diffusing away. So it functioned. But what we're interested in from the academic perspective is that we got a human implant back. How often do we get a biomaterial that we've implanted in a person back, right? That's a unique opportunity, so we tried to make the most of that. What we did notice is that the response to that material, if the response to that material was different when it was near dermis or subcutaneous muscle or adipose. So you could take the same biomaterial and put it just in different tissue environments and get very different responses. So this led to actually me going on a sabbatical and trying to learn some immunology a couple years ago. We also developed a technology that allowed us to create essentially different tissue environments on a chip. So we could take any organ that you want, so lung, liver, spleen, bone, whatever you want, and we process it so we just have that scaffold extracellular matrix that is there talking to the cell, and we could print that on these arrays. We could have them in 2D or in 3D. And we could screen a lot of different stuff. So we put a lot of different cell types on there. One of the questions we got from reviewers, this is in press now, was does it really mimic the natural environment? So we went to our proteomics friends and said, what's in these mixtures? Does it really mimic the native tissue? And it mostly does. And you can play around with the processing to create these different tissue environments. I do have another stem cell slide. These are adult stem cells that we put on this tissue array. And we looked at osteogenesis, and we could screen, and we got some unexpected things, like lung really supported osteogenesis. Why would that be the case? Well, again, we were sent back to do some more work by the reviewers, and we went to a systems biologist. I've always wanted to work with systems biology and big data and try to be more quantitative in regenerative medicine. But it was pretty hard. We didn't really have a lot of data. We tried to make these tissues in a dish, and it was too laborious to get big data. But here we had a lot of proteomics for what was in each of these tissues, and then we could screen all these different outcomes. So we did find a connection with gene ontology analysis looking at that composition of the lung proteome. And if we looked at elements related to bone formation, actually lung did have a fair amount compared to the control spleen that did not show any benefit in osteogenesis. So we can start teasing out those different factors that can be responsible. And lung calcification in cancer is something that is well observed. So then we looked at screening different cancer cells, adhesion and proliferation on these different matrices. We could look at parent versus metastatic lines with the melanoma. What do they like to stick to? Okay. We also went back to the immunology that I said I was trying to learn something about and looked at how macrophages responded to these tissue environments. So again, a lot of screening. The main point I want to share with you is that we can start making connections. Right? So you have that same scaffold environment that is influencing things like stem cells, regeneration, cancer interactions, and immune responses. Are there connections? So again, we went to our systems biology friends and put all these different assays together. You don't need to read that. It's just all those different cancer, immune, stem cell differentiation assays, and then connected it with proteins. Made all these different connections, and you can start making a tree. So what are the connections to a positive or negative response with a macrophage or a stem cell or a cancer cell adhesion, and start picking out these proteins and starting understanding what elements of the message that extracellular matrix is guiding that local environment and the cell response. So, and the last thing I'll mention with tissue environments is we are working on translating them. So that tissue array, we're taking the element of adipose and putting an off-the-shelf adipose that we've processed. And this is in collaboration with the Armed Forces Institute of Regenerative Medicine. So doing a little bit like what Kythera was trying to do, except for larger defects, and using those tissue environments to direct repair. Now, the last thing I'd like to spend a little more time on is getting back to that scaffold environment. But instead of talking about stem cells or fully differentiated cells, talk about immune cells. I'm sure no one has any interest in immunology here. But we've got a lot of different cell types that might respond to a biomaterial environment, right? So I've got macrophages, T cells, dendritic cells, B cells, everything in that environment. And what you saw from that data in those tummy tucks is that these are really the first responders, right? So we can use a biomaterial to help direct these first responders. So a few key cell types that we were interested in looking at were the macrophages that come in there, right? And so those macrophages can be involved with debris clearance. They can also be involved with antigen presentation and also with regeneration, depending on their phenotype. Dendritic cells are living in that tissue and can be active in presenting signals. I'm not going to talk too much about neutrophils, but they're definitely important too. And then, of course, these elements of the innate immune system are talking to components of the adaptive immune system. And we're going to be talking mostly about the T cells here in our responses that we see. Now, I mentioned I like quantitation, right? So I was excited about working with a systems biology person and getting big data. And we can do that now with immunology. We're working with some friends in cancer immunology, and we feel very lucky because they've got lots of cool techniques, right, that we can start using. Tissue engineering and scaffold biomaterials development used to be a black box, right? We used to say, okay, we put a scaffold in there and see what comes out. We really don't know what happens, though. Now we can start dissecting out every single cell that comes into these implants and try to select them out and understand what they're doing, right? So we have a great student here that I show hiking because I think she's definitely being a trailblazer with this. She's had some immunology background and so can bridge the engineering and the cancer immunology lab in a very nice way. And we're starting off with just a simple muscle trauma. So this is a mouse muscle, taking out part of the quadriceps and putting in different materials and saying what comes in there, right? So particularly interested in these macrophage populations here. and these T cell populations here that are coming into the defect, and what happens to them over time. Well, when we just take that whole leg and look at what's going on in that muscle, we see some interesting genes being expressed. IL-4 and various other, what do we call, Th2 responses that are more of, let's say, regenerative response in this case, at least I hope to prove to you. And we see that happening, this expression in wild-type normal mice. When we take a mouse that doesn't have T cells or B cells, we lose that pro-regenerative environment that's created by this, these markers. So we look at it a little bit closer. If we just look at the macrophage population here, we're just looking at macrophages, and we're going to look at the marker CD86, this M1 marker, and CD206, an M2 marker. And what's interesting is, you know, in a plain saline defect, you do that trauma, and we inject some saline in there. There's no difference if you have the wild type or the mouse that's lacking those T cells and B cells. However, we first notice in the wild type that the expression of those M1 CD86 markers goes down. But if we don't have those T cells around, we don't have the benefit of that biomaterial in decreasing that sort of pro-inflammatory macrophage. Now, if we look at CD206, this M2 marker, well, with no treatment, the saline, and with one of our extracellular matrix environments, we don't see too much of a difference in the number of CD206 cells, except when we lose those T cells or B cells. So these T cells are really important in helping to instruct the macrophages to be more of an inflammatory phenotype or more of a reparative phenotype. How does this relate to actual muscle regeneration? Well, as we were dissecting out, again, a great student was able to notice, hey, there's some lymph nodes that are really big here. So she grabbed these lymph nodes with treatment. You see it's much larger than without treatment. And if you look at expression of some of these markers, again, this IL-4, and it wasn't just the lymph nodes that were close by to that defect. Also, distal lymph nodes had those changes in that expression. So a locally implanted biomaterial can have some systemic effects in the immune system. Now, what does this mean for actual functioning? Why should you care if there's more IL-4 expression or this immunomodulation? Well, here's our wild-type mouse and the repair that we're seeing with treatment of our biomaterial. And from muscle biologists have told us that these are good things with these nuclei here and the size of the muscle fibers compared to without T cells. You see they're small, not really repairing. We also see increased adipose when we don't have the when we don't have those T cells. Now, functionally, though, that's just a nice picture. Functionally, what happens? Well, we put these mice on a little running regimen, and what we find is, you know, here's our healthy mouse with and without T cells here. As we incorporate biomaterials, we can increase their rate of recovery, increase their level of recovery from these defects. But look here, this red, again, without the T cells, we don't get the functional recovery. The animals are not allowed, they are allowed. They aren't able to run the same way as those cells that have that intact immune system and those pro-regenerative signals from the scaffold. So you can start thinking about applying this to any tissue environment. That's what happens in the muscle environment. But if we go to a knee defect, even a neural defect, skin defects, what does that local tissue environment look like from the inflammatory perspective, and how can a biomaterial modulate that? And we all know now that T cells are very popular targets in things like cancer immunology. So now there may be going to be important targets for another area of what we'd like to call regenerative immunology. So when you have an injury, a lot of things happen. A lot of immune cells are activated. Neutrophils coming in first. If we can create an immune microenvironment with these biomaterials that helps direct them to a more pro-regenerative phenotype, we can start thinking about moving more quickly towards a regenerated tissue instead of a scarring or a chronic wound environment. What I'm very excited about is also collaborating with transplant immunology folks. And I think this is an important triad here that will really have some important future impact. We've been working closely with cancer immunologists in all of this. This is, I don't want to say it's a fully mature field, but there's a lot going on and it's pretty mature. Transplant immunology has also been around for a while. A lot of exciting things going on that we can do all the amazing transplants we do. And we've got some groups at Hopkins that are working on face transplants and hand transplants. Interestingly, these two haven't really talked to each other. The communities in general haven't really spoken to each other too much, but there's a lot that can be learned. And I think through the connection with regenerative immunology, we're going to bring all these people together. So a few themes that I hope I've been able to get across for what's going to be happening in the future with regenerative medicine. I think we're going to move from an experimental empirical science to be more quantitative, actually get data so we can start being a little bit smarter about our design. And hopefully this design will be connected with therapeutic outputs. Right? We really need to understand what are the key design factors that will impact therapeutic outcomes. And I'm hoping systems biology can play a role in that. The other aspect that I hope to see is the leveraging of the immune system to help us in this regenerative immunology. And finally, translation with academic-private partnerships, particularly as we're transitioning from the discussion over the past 2 days with more private or industrial activity and moving into science, basic science talks tomorrow. This concept of the academic-private industrial partnership is really key, and you see universities all over the country trying to enhance their ability to work with partners outside in this. And also, it's a real opportunity for faculty to learn about translation of their technologies. And again, I'll say it one more time, try to connect more with therapeutic outcomes. So what we're studying in the lab does have a connection to what can happen in improving patient care. So there are many people to thank, collaborators. This is just one picture of our group. It's actually an old picture of the group. And I just stuck in a new picture of the group, which I think represents the toughness of the group now. And it is paintball, not real, not real ammunition, but I've been told it really hurts. I declined to be a part of it. I either figured they're not going to shoot me at all, or I'm going to be a real target. So either way, it wasn't a good thing. So I went over a lot of different topics rather quickly, so I would be really happy to answer any questions more in depth about any of those areas, and then we'll be just in time for the sunset. So thank you for your attention. No questions? I put you to sleep? Or I'll get a question after a glass of wine.
What kind of T cells went into the, or attracted by the biomaterial? Did you get regulatory T cells, or Th2 cells, or?
Yes.
And how do you deal with IL-13 from Th2 cells, which is profibrotic?
Yes, so we actually sorted the cells. So we sorted out the T cells and the macrophages, and the Tregs, actually, we did see a little bit of an increase with the materials, but not a huge increase. And as far as expression, we actually saw pretty significant IL-4 expression from the T cells themselves. And we also saw some, was it Th17 stuff coming up, which was a little bit unique. So I have like 50 genes there that we looked at. So I don't remember all of them, but those were the two that we're sort of targeting on. So now we're doing experiments on looking at the CD4 cells. It's probably the CD4 cells that are the biggest impact. So the materials actually were decreasing the CD8 cells. And there are some there's some studies out there in bone regeneration that show how differentiated CD8 cells were blocking bone regeneration actually in patients, and then in the model was a very nice study. So we're going to be targeting more of the CD4 cells as far as probably the ones that are implicated, but I think the Tregs are very interesting, and the work with the transplant folks are key. And what's interesting is that the biomaterials people are just starting to get involved with immunology, and it's sort of like, well, you don't want M1, you want M2, and actually that's really not true. It's much more complex. And what we're seeing is that the macrophages actually have a fair amount of the MHC class II, and are probably doing a fair amount of antigen presentation. So I think that that sort of M1 phenotype early on is probably necessary, and then shifting to more of the M2 Th2 response We haven't seen it go over to being fibrotic yet, but I actually would love to get involved with some animal models where it does become too fibrotic, and we can see what the biomaterial does. When we looked at the different tissue environments, what we saw on the tissue array was that when we culture the macrophages in sort of a extreme M1 or M2 media environment, the biomaterial seemed to moderate it a little bit. So it wouldn't be as extreme. So that's just another piece to throw in there.
What is the immunogenic target of the T cells? And is that the biomaterial itself?
We don't know yet. Now that's the complex, we've looked at synthetic materials, but what I presented was just the biological material, so it's a complex mixture. So that's a tissue-derived biomaterial, and what we're looking at is where do you see differences in the different tissue environments? So when do you start, and then when do you start losing the tissue specificity? And what I'm trying to say with that is we've also made nanoparticles with those different same tissue environments and see them get tracked to the lymph nodes. So we are now doing a screen for trying to look at sort of the clonal proliferation of the T cells and what actual antigen targets they're looking at. So we've got a screen that we submitted to a company. So hopefully I'll have an answer to that soon. Yeah, they are reacting to the material. That's what we think. That's what it looks like. Yeah.
Actually, it went pretty fast, so I did not catch it. So partly you talk about matrix for bone repair, and partly talk about cells for bone repair. So regarding the bone repair, do you think a matrix is more critical than the cell, or have you tried any combinations?
That's a great question. So I think engineers often like to over-engineer things, and so I think we oftentimes don't respect what the cell can do. And so particularly also with the translation process, you want to simplify things. So sort of simple engineering that's practical to go into people. And so I gave 2 examples of things that went into people. And the third example that's going to be starting, hopefully we're manufacturing starting tomorrow, is with the extracellular matrix. And we were unhappy with something the FDA told us, but it's actually going to be good. So our first trial was actually supposed to put those matrices into defects, soft tissue defects in people. But they said, okay, we want you to actually take the implants back, sort of more like the Kythera abdominoplasty. So we're going to get the implants back, but now that we've got these immune techniques set up, I'm going to be able to then do the same thing that we did almost in mice. I'm going to be able to do it in people with that one particular implant and start trying to make some correlations of, well, does that mouse immune data make sense, and is that what happens in people? So I think we can bridge it. It's going to be much more simplified, the technologies we use for clinical translation. And there's a lot of great stuff going on right now for manufacturing cells. And when somebody else figures that out, I'm happy to put it with our materials. Yeah, there's a lot in that question. So I was really excited to see, there was a paper that came out of Stanford about, I think, a year and a half ago that looked at taking, they were doing single cell analysis of blood with patients undergoing, I think, a knee implant. And they were able to correlate sort of immune signatures that correlated with a patient that had a 10-day recovery or a 10-week recovery. Right? So that makes you start thinking that, okay, there's a trauma of the surgery, right? And the systemic immune state of that patient impacts the response to that surgery and if they're going to have a 10-day or 10-week recovery. And the same patient could have a 10-day or 10-week recovery depending on their immune state. So I think there's a big potential impact for that, just even for simple surgical recovery, much less sort of traumatic wounds or disease processes. I think these things might be happening anyway, and we don't know it, right? So this is also partly a discovery. I think it makes sense that you would have systemic immune changes when you have a local trauma, right? So I don't think we're necessarily changing that. Biomaterials have been used in these applications in people before, so I don't think we're changing anything, we're just understanding, so then we can start leveraging it. And again, we're then taking these scaffolds and making them into small particles then, so then you don't even have to put in a defect, you just have to inject it, and it's actually moving towards the lymph nodes. So we're trying to understand all of that. We haven't done the full analysis of the lymph nodes. We're in the process of doing all those now, but we look forward to seeing those results. Any other questions? Okay, well, perfect timing at 6:24. Thank you.